No. Hear me now? Okay. Okay, thank you. Okay, thank you. Well, good morning. I hope you all of you have logged in. You have listened to the application, and prior to that, you have seen the class on the structure of DNA and RNA. Now, one important thing which I want to specify over here is fidelity of replication is maintained. Now, fidelity of replication is maintained by Chargaff. So you have seen all of these. It is very important that the fidelity of replication has to be maintained throughout the life of the cycle. Now we know that replication takes place at the time of a cycle, and therefore it has to be high. In order to conserve the DNA molecule, there are of mechanisms which are responsible for maintenance of the structure of the DNA. Okay. Now, before we proceed further, we should know something about we will just revise briefly types of polymerases. Now, DNA polymerases, as you know, are the major enzymes which are involved in the synthesis. We have got three types in the case of E. coli. There's 1, 2, and 3. Now, all of this polymerase 2 and polymerase 3. The functions of these polymerases is called as and repair. DNA polymerase 2 has got this inherent quality of proofreading. Now, what do you understand by printing? We will first put on all the words on the printer and then take out a copy One is the decarboxylase enzyme. Then there will be, then again, of course, once the base is removed, the new base or the complementary base will be inserted to DNA polymerase. And then finally, DNA ligase will put it together. Mechanism, the nucleotide excision repair. Now, this mechanism, as the name indicates, there is removal of a bigger chunk. A number of nucleotide bases will be excised from the newly synthesized DNA, carried out by a specific type of a enzyme which is called as an exonuclease. It is different from the exonucleases and the of prokaryotes. This enzyme is called as an exonuclease. And a similar type of an exonuclease enzyme is also found in the higher organisms. The damage may cause large structural changes. For example, formation of pyrimidine dimers, which have to be removed. Carried out by removing around 30 nucleotides, and that will be replaced. Okay. Then another damage which can be carried out is breaking of both the DNA strands. Both the strands of the DNA, the replicated DNA, will break off. So that is called a double strand break. Now, for double strand break, usually is because of ionizing radiations, chemotherapy, oxidative free radicals, etc. Now, this is a very unique mechanism which involves a number of steps. Each of these steps I will be dealing with shortly. Okay? Now, the first one, mismatch repair. As the name itself indicates, a wrong base has been inserted. It is not matching the base that is present in the parental DNA. So, in order now, the enzymes, the repair enzyme between the parental strand and the newly formed strand. Supposing this is our parental strand, right? These two, this is the parent DNA molecule. Now, what will happen is the parental strand will be methylated so that it can be distinguished from the newly formed strand. So, this is the newly formed strand. wrong pair, and it has to be removed. How it will be removed? First of all, at the GATC sequence, the strand has been methylated, parent strand is methylated at one or more positions. Fine. There will be a GATC endonuclease enzyme which will form a cut near the mismatch site. Okay. Now this cut will be carried out by an endonuclease enzyme. You know, endonuclease will cleave the DNA molecule within the molecule. Okay. Now once this happens, in the 5' to 3' direction, an exonuclease activity will now take place. Now this exonuclease activity will start removing the whole of this nucleotide sequence up till here, say up till here, which is the mismatch area as well. So first thing, endonuclease will form a nick. Endonuclease forms a nick. Then exonuclease will start digesting this whole nucleotide sequence, which will include this mismatch site as well. So a whole sequence like this, including the mismatch sequence, is now digested by an exonuclease enzyme. Fine. Now, once this exonuclease enzyme has removed this complete set of nucleotides, this defect will now be repaired by the DNA polymerase enzyme. So once the DNA polymerase enzyme will now put in the right base, matching has taken place, the right base will be inserted. This wrong base which was here will now be repaired, and then the gaps will be again filled in by a ligase enzyme. Okay, and the newly synthesized DNA without the defect will now be clear. So once again, a GATC sequence is recognized. Then near that GATC sequence, the adenine molecule, it will be methylated so that the parent can be distinguished from the daughter strand, right? Once that is done, a GATC endonuclease enzyme will nick. At this nick, the exonuclease enzyme will now start acting. The whole of this molecule will now be digested. This is now digested. A new is inserted by a polymerase enzyme, and religation will take place by a ligase. So in this way, the mismatch repair is now taken care of. Now, since of the parent strand takes place over here, this type of a repair mechanism is referred to as a methyl-directed mismatch repair. Now, supposing there is some problem with this type of a mismatch repair, it can result in specific disease which is called as HNPCC, or hereditary non-polyposis colonic cancer. Now, this is because of a defect in this gene, that is HMSH2/1 on the chromosome 2. More of this later on when we talk about the diseases associated with the repair mechanisms. On to the second mechanism, that is base excision repair. Let us figure carefully. This is the figure, okay? Now this is the normal base, and C is the complementary base pair. Now normally, as the name indicates, base excision repair means a wrong base has been inserted during the replication process. Now, which are the wrong bases which can be inserted over here? For example, cytosine may undergo deamination to form uracil. Adenine may undergo deamination to form hypoxanthine, and guanine can be converted to xanthine. So instead of cytosine, adenine, and guanine, wrong bases like uracil, hypoxanthine, and xanthine, they can be inserted. Now look at this. Now usually due to exposure to some external phenomena such as heat or UV, etc., what will happen, instead of cytosine which was here, see now this is cytosine molecule, if this cytosine is now converted to uracil, okay? This cytosine is now converted to uracil, okay? Now once this has taken place, what will happen now? This uracil has to be removed. Now this uracil is removed. this uracil is removed by a uracil DNA. Now, as I had told you in the beginning, the enzymes that are required for this are a DNA glycosylase, then a nuclease, and finally the polymerase and the DNA ligase enzyme. Now, DNA glycosylase, as the name itself indicates, it is an enzyme which will act upon the glycosidic bond. That is the bond between the ribose sugar and the nucleotide base, the nitrogenous deoxyribose sugar. Okay. Now, this detail I will come to. So, first, the DNA glycosylase, it will recognize a damaged base. Okay. So, this is the damaged base over here. This is the damaged base. It will recognize and it will cleave between the base. Okay. So, now, remember in the methyl mismatch repair, a chunk of the nucleotides are removed from the newly strand DNA. However, in the case of base excision repair, it is not so. Here only a single base will be removed. And it will cleave this base and deoxyribose from the backbone of the DNA molecule. Now, the particularly common DNA lesions are products of cytosine and adenine, which I have already told. Once the glycosylase removes the base, what will happen? It will result in formation of a apurinic. This should be not apurinic, it should be apurinic. This A will be here. And apurinic or a pyrimidinic site. That is a site where there is no purine or pyrimidine available because it has been excised or taken away, and that is here. So the uridine molecule has been removed from it, and this has created an AP site. This AP site may be either removal of a purine base or a pyrimidine base. Okay? So once this is done, the DS will now cleave off the deoxyribose sugar as well, and DNA polymerase will initiate the repair or synthesis from the free hydroxyl end of the link. The damage will be removed, and this it will be sealed by the DNA molecules. So this is carried out. So now this is what happens. Okay? Nucleotide excision repair. This is what happens. Now this is the newly formed DNA. This is the parental DNA. Now, primers or a DNA lesion has occurred. Wrong base has been formed. There is a modification of the base. Once this happens, a large distortion can take place over here in the DNA lesion. So, in nucleotide excision repair, there is a slight difference. What is the difference? A single base, there will be removal of a large base. Okay? So DNA lesions are usually large distortions which can occur in the structure of the DNA molecule. Now there is an enzyme ABC-XZ nuclease. This ABC-XZ nuclease is found in the prokaryotes. So why it is named as ABC-XZ nuclease? Because it contains 3 polypeptide chains, each of which is a product of 3 different genes, UVR-A, UVR-B, and UVR-C. That is why it is referred to as a ABC-XZ nuclease. This ABC-XZ nuclease is responsible for the cleaving off of the whole DNA. Will cleave off. In the case of eukaryotes, supposing this is our damaged strand, okay, it will cleave the damaged strand at the 8th phosphodiester bond, 5' side. This is the 8th phosphodiester bond from here to here. 8th phosphodiester bond on the 5' side of the lesion, and the 4th or 5th phosphodiester bond on the 3' side. So totally around 12 to 13 base pair region is simply cleaved off. So a big nucleotide sequence is cleaved off by an XZ nuclease enzyme. In eukaryotes, In the case of higher organisms, there is a difference. In human beings, the cleavage portion will be the 22nd nucleotide on the 5' side and the 6th or 5th one on the 3' side. So a chunk of around 27 to 29 nucleotides, which will of course include the lesion, this DNA lesion, it will be cleaved off. Fine. Once this whole nucleotide sequence is cleaved off, the repair will be carried out again. We will now fill out the whole gap. This whole gap will be filled out like this and it will be repaired by the DNA molecule. Now the resetting or gap will be filled by DNA polymerase and it will be sealed by ligase. In the case of DNA polymerase, delta as well as the epsilon. So once again, the difference between and the nucleotide excision repair. In the case of base excision repair, what we had seen was there is a removal of a base directly. Okay, this base was removed directly first by a glycosylase enzyme, then a nuclease enzyme removed the deoxyribose sugar to create an apyrimidinic site. So in this case, the nucleotide molecule was not broken away from the new strand. Only a single base was removed and it was simply excised away and then it was sealed by DNA ligase. Whereas in nucleotide excision repair, what we are seeing is a big chunk of the nucleotide has been removed, which includes the DNA lesion, maybe a thymine dimer or any such, which is induced usually by the external factors. Okay? And once again, the two enzymes, DNA polymerase and ligase, will come into the picture, and the damage will be nicely repaired like this. Okay? Now, a number of diseases associated with this nucleotide excision repair. One of them is the xeroderma pigmentosa. I will come to it later on. Now we move to the fourth mechanism, that is the double strand break repair. Now, double strand break repair, as the name itself indicates, there is a break in both the strands of the DNA molecule. See, you can see there is a break in both the strands of the DNA molecule. Now, if this happens, these two strands, they cannot come close together. We know that in order for these strands to come back together, they simply cannot unite like this. There has to be again complementary base pairing between the two strands by formation of sticky ends, so that base pairing can take place. Now, this is a very important phenomena. Now, usually double strand breaks can occur during a process called as gene rearrangement. Gene rearrangement is a form of regulation of gene expression where a number of antibodies can be synthesized upon exposure. Now, we know that whenever we are exposed to any external molecule, a large number of antibody can be synthesized against each of these external molecules. Now, we do not have that many genes for the synthesis of individual antibodies. So how we are able to synthesize thousands and thousands of antibodies against to any external factor? This is done by a process of gene rearrangement. The various elements in the genes will rearrange themselves and that will result in formation of different antibodies. This you will be studying in the regulation of gene expression. So usually what I want to stress here, during the physiologic process of gene rearrangement, double strand breaks can occur like this. Okay? Now, once double strand break occurs, a mechanism for the repair is referred to as a double strand break repair. Now, this double strand break repair, now this is a slightly complicated mechanism, so you'll have to listen carefully. There are two major proteins involved, a Q protein and a DNA dependent protein kinase. Okay? Now, this Q protein two subunits. These two subunits are of 70 kilodaltons and 86 kilodaltons. So whenever damage occurs like this, a double strand break can occur either due to ionizing radiation or due to exposure to chemotherapeutic reagents or due to exposure to oxidative free radical generation. There will be such a break. Okay. So now these two proteins, which has got two subunits, and a DNA dependent protein kinase. Both of them will come together and they will bind to the double stranded DNA. Now this DNA protein kinase has got a very unique feature. What it will do is, it has a binding site for the DNA free end. It has got a binding site here for the DNA free end. And it also has a binding site for the inside portion of the double stranded DNA. Okay. Just inside the end where the break has occurred. Okay. And the Q protein has got an intrinsic helicase activity. You know what is the helicase activity? The helicase activity is responsible for unwinding of the DNA. So once this dimer is formed, the Q protein and the DNA protein, the DNA dependent protein kinase, they bind to the double stranded DNA. A structure like this will be formed. There will be approximation. The two DNAs, DNA PK or the DNA dependent protein kinase, it will bind both to the Q protein, to the inside of the DNA, to the inside of the DNA molecule, as well as to another DNA PK molecule. Now, due to this binding, the protein kinase activity of the DNA PK will be activated and phosphorylation will take place. Once phosphorylation has taken place, there will be approximation of the two such strands, one about the other. After this phosphorylation, what will happen? The activity of the DNA helicase activity of Q protein will be activated. Two strands will now open up like this. The two strands, this double stranded molecule, this molecule, they will now open up like this. And the DNA protein kinase, it will now separate. So unwinding of the DNA will take place first by phosphorylation of the DNA, phosphorylation by the DNA dependent protein kinase. The two big parts of the DNA which approximate about each other. Then the DNA PK will be removed. The Q protein helicase activity will be activated and unwinding of the DNA will now take place like this. Once this has happened, these are the two loose ends. Now this DNA and this DNA strand, they both will now align themselves. They will align themselves like this, and the complementary base pairing between these two will take place. So this is how the complementary two strands. Now these are the loose ends. Now these loose ends, they will be cleaved off by nucleases. Now complementary base pairing has taken place. These loose ends from the two strands, they are now removed like this, and after ligation, this double strand break will be repaired. So this is how the double strand break repair occurs. Now this is a little complicated. So let me explain once again. There is a double strand break due to exposure to damaged DNA, ionizing radiation or oxidative free radical generation, or maybe exposure to chemotherapeutic reagents. Then two different types of proteins, a Q protein which is a heterodimer and a DNA binding protein kinase, they will bind to the double stranded DNA. Now this DNA protein kinase, it has got two binding sites, one for the double stranded DNA and another for binding towards the inside. So now we have a complex of Q protein, DNA protein kinase bound to the double stranded DNA and also to the inside of the DNA, so that the kinase activity is now activated and phosphorylation of the DNA will take place here. Once phosphorylation takes place, there is approximation and the broken strands, they are lying one above the other like this. As soon as this complete binding takes place, the DNA protein kinase, double stranded DNA binding resulting in the protein kinase activity, phosphorylation of this, and results in an approximation. As a result of this, the helicase activity of the Q protein will now be activated. And the two strands, these two double strands here, they will be unwound. Unwinding will take place like this. Unwinding has taken place. What will happen? These two strands, the parental strand of one DNA and the second complementary strand, the loose ends will come together and there will be, there will be alignment and complementary base pairing will take place. These two loose ends, now they will be nucleases and ligase will now repair the damaged ends. Okay. We come to the clinical aspects. The diseases due to the defects in the DNA repair mechanisms. As I had told you in the beginning, the mismatch repair. Now, this mismatch repair, if there is a defect in a particular, it will result in a disease called as hereditary non-polyposis colon cancer or HNPCC. Now, this particular disease, there is a defect in at least 5 different mismatch repair genes. For example, if there is a defect in the MLNH gene, the human mute and HMSH2 or human muteus homolog 2 genes. So whenever there is a defect in this, the mismatch repair will not take place and it will result in a specific type of cancer that is CC. Now this cancer develops at a very early age. Now this is a recessive genetic that occur in different families. Okay. Then I have told you, spoken about the nucleotide excision repair. A very important disease that is associated with repair is pigmentosum. Now, xeroderma pigmentosum is unique to. Now, you must have observed that many a times what happens is this again, xeroderma pigmentosum. It is an autosomal recessive genetic disease. Now, the clinical syndrome here includes a marked sensitivity to sunlight. So whenever the person goes out in the sun or exposed to UV rays, there will be formation of, say, lesions on the skin. Thymine dimers may take place. Now, if the nucleotide excision repair enzymes are in place, these dimers will be immediately removed by the nucleotide excision repair. But if these enzymes are defective, then it will result in multiple skin lesions, which can then lead to cancer. So this is a very serious disorder. So the inherited defect involves the repair of the DNA damage. Particularly, the damage is due to thymine dimers. Excision repair is a disease when these enzymes I have not mentioned over here. Let's see. Okay, I'll just tell you here. Okay, ataxia telangiectasia. Now, this particular disease is again autosomal recessive disease in the human beings, and this results in the development of cerebellar ataxia and liver neoplasms. And there is an increased sensitivity to damage by X. Now, this is not exactly a defect repair enzymes, but because the repair enzymes are also involved in some other mechanisms. For example, in the transcription process and in the cell cycle process. So together, the person will become sensitive to damage by X-rays. Okay. Then there is another syndrome, Fanconi's anemia. Now this is also an autosomal. It is also an autosomal anemia. This is characterized by an increased frequency and by chromosomal instability. So Fanconi's anemia is again an autosomal recessive anemia. Autosomal recessive anemia, and it is characterized by an increased frequency of cancer and by chromosomal instability. The defect is in the repair of cross-linking damage. So I have not damage defect. We are only going to study 4 such defects. Okay. Then, now we have got some more diseases. One more is the recombinational DNA repair. Whenever recombination of the DNA takes place, it can result a defect in the breast cancer 1 or breast cancer 2 genes in women. If human breast cancer, women with a defect in these 2 genes have maximum chances of developing breast cancer. And this development is due to a defect in the recombinational DNA. Okay. So we have briefly revised all the 4 mechanisms. Now we come back to our original table, where now you will be able to understand what are the different mechanisms. So what we have learned today, I will just briefly summarize. A number of mutations can take place or damage to DNA can take place. This damage may be due to single base alterations such as depurination, or it may be due to deamination of cytosine to uracil, or it may be due to deamination of adenine to hypoxanthine, alkylation of different bases, etc. Then there may be a 2-base alteration. On exposure to UV light, thymine dimers can form. Then there can be even bifunctional alkylating agent, cross-linking damage may take place. Then sometimes chain breaks may take place, double strand breaks may take place due to exposure to ionizing radiations, or due to radioactive exposure, or due to damage by oxidative free radical generations. Then double strand break repairs can also take place. The 4th one is cross linkages repairs. Now, in order to combat different types of DNA damages or mutations, we have got 4 different mechanisms. One is the mismatch repair, then base excision repair, nucleotide excision repair, and double strand break repair. Now, mismatch repair is taken care of by a set of enzymes, namely the various mutation proteins. In the case of prokaryotes, they are MutH, mutal, and mutase. Analogous proteins are also found in the eukaryotes. The major enzyme here is a DAM methylase. Now, this methylase is responsible for methylation parental DNA. So, first of all, there will be methyl-directed strand cutting. The methylation will take place at a GATC sequence near the damaged site, so that the parents identified from the daughter strand. Once this takes place, the endonuclease will give a cut near the GATC sequence. Okay, and Then the exonuclease will now remove the whole of the nucleotide strand, including the damaged portion. The new portion will be now synthesized by the DNA polymerase, and then it will be ligated by DNA ligase. Okay. Then the base excision repair. Now, in the case of base, a number of enzymes are involved. DNA glycolases, then apurinic or apyrimidinic endonucleases, then of course the DNA polymerase 1 and DNA ligase. Now this base excision repair is for the removal of abnormal bases. Now how the abnormal bases, which are the abnormal bases normally found? They are uracil, hypoxanthine or xanthine. Now in this case what happens? First the glycosylase, I will just show you the figure once again. First the glycosylase. Now this is the wrong base inserted. First the glycosylase will remove that base. Once the nucleotide base, say uracil is the wrong nucleotide base, it is removed by the glycosylase. Once this is removed, an apyrimidic site will take place. The nuclease enzyme will now cleave off the deoxyribose from the backbone. Now please note here, the DNA backbone structure, it is not disturbed. Only the base is excised from here, and the nucleases will now create this AP site. AP site is formed. The DNA polymerase will initiate the repair. The right base, that is again cytosine, will inserted over here, and this right base will now be taken care of by the DNA polymerase, and the DNA ligase will now repair this. Okay. The nucleotide excision repair. Now, in case of the nucleotide repair, we have found that this takes place due to formation of DNA lesions. A number of DNA lesions can cause large structural changes. For example, thymine dimers in the case of this. And nucleus. This is a 12 to 14 base pair nucleotide will be removed from the, which will include the lesion also. Now I have to show you. See. So this will remove. So ABCXC 12 to 13 base nucleotides. And in the case of human beings, the enzyme which is into ABCXC nucleus is much more larger. It may contain more than 16 polypeptide units. So it is a very big molecule. And in this case, the nucleotide which is 22nd on the 5' side, this is the 5' side of the nucleus, and 6th nucleotide on the 3' side. This whole thing, which included the DNA lesion, it will be simply excised from the DNA, and once again the DNA polymerase and the DNA ligase, they will bind them together, and this nucleotide repair will take place. So a big chunk of the nucleotide is excised and then repair takes place. So in base excision, we are not disturbing the backbone structure. In the case of nucleotide excision and in the case of the backbone structure of the DNA is also disturbed, and a chunk of the molecule is removed, and then repair is taken place with the help of DNA polymerase and DNA ligase. Then the Fourth one was double strand breaks can be because of exposure to ionizing radiation, or it may be due to chemotherapy, or it may be due to oxidative free radicals. Okay. Now this process is a little complicated. So I have already repeated it to you the major steps. See, proteins involved are a two protein and a. First of all, this DNA protein, they will come and bind to the double stranded DNA. This has got a unique ability. It can bind to the 2 protein. It can bind to the of the double stranded DNA, and at the same time, it also molecule of DNA protein kinase. Once this happens, the protein kinase activity is phosphorylation occurs. The 2 strands will now be occurs. The 2 protein will be activated, and unwinding of these 2 double stranded DNA will take. That's unwinding, alignment, and complementary base pairing between these 2 opposite strands will take place like this. This is the base over here, and these loose ends, without any base pairing, these loose ends, they will be cleaved up from the, and ligation will take place, and the double strand break repair will occur. We come to the diseases due to DNA. Whenever there is defect in the mismatch repair enzyme, now this is again a hereditary disease. There is a defect in at least 5 different mismatch repair enzymes. For example, in the MUT H, M, L, and H. In the human case, the analog is MUT L homolog 1, and this is the one in the case of prokaryotes, and in the humans, it is the human MUT S homolog 2 genes. Now, in individuals which this type of defect in the repair enzymes results in a disease which is called as HNPCC, hereditary non-polyposis colonic cancer. This will develop at a very early age, and colon cancers are most common. Then, when there is a defect in the nucleotide, I have already told you, there will be development of mostly thymine dimers on the skin, and these cannot be repaired. Therefore, the person becomes very, very sensitive to exposure to sunlight and UV rays, and this will, whenever there is such exposure, it can result in skin cancer. So, 2 major diseases. One, due to a mismatch repair enzymes, that is HNPCC, due to a defect in the nucleotide excision repair enzyme, xeroderma. This should be xeroderma. There's a mistyping mistake over here. It should be xeroderma pigmentosa. Now, in addition to this, besides the repair mechanism, some of these repair enzymes, they are found to be functional. They are participating in several other activities as well. For example, some repair enzymes are also found in the transcription factor 2H complex, and it can play a role in gene transcription. Then another component of TF2H is also involved in the cell cycle regulation. So when there is such a multifactorial, different diseases can occur. One such disease is extreme sensitivity to exposure to X-rays, which is a autosomal recessive disease called as ataxia-telangiectasia, discussed earlier. Then, whenever there is a defect in recombinational DNA repair, it will result in development of breast cancer, where the defect is in the breast cancer 1 of women. Then, if there is a defect in repair of cross-linking damage, and there is a defective repair, it can lead to Fanconi's anemia. Now, Fanconi's anemia is autosomal recessive anemia, which is characterized by increased frequency of cancer and chromosomal. So, with this, we complete the various repair mechanisms and the defect diseases due to the defects in the DNA repair mechanism. Okay, now I will end the class. I hope you have understood. If you have any questions, you can ring me up on my phone number. I'm announcing it. For those of you who are interested, you can just note it down. If you have any problems, I do not know when the college will open up and when we are going to meet. So anyway, you just jot down. If you have any problem, you can talk to me with this particular topic. 9818 39 7601. Okay. So till then, bye-bye. Take care of yourself. Be at home and take all the necessary precautions. And I hope whenever the college opens up, we meet again. Okay? I'll be taking your next class on transcription day after tomorrow. Okay? So please be alert. The link will be sent to you once it is created. Okay? Thank you. Bye-bye.